University Medical Center. It is my pleasure to introduce to you Ashwari.
Serious? Oh my God, you could have said, what happened to her? Oh my goodness.
My name is Ashwari, and my research focuses on the relationship between total receptors and NF-kappa factors is dependent on genetic identity and physical structure. Total receptors are proteins which induce a process known as cell competition in order to protect our immune system. Cell competition is a process which eliminates cells which are harmful to our bodies. And in order to begin this process, the receptors must first send a signal to the NF-kappa factors. NF-kappa factors are a type of pathway which induce this process. Within the NF-kappa factor family, there are many types of factors, such as REL-A, REL-B, REL-68, and so on. This then activates a pro-apoptotic inducer, which induces a process known as apoptosis, which is cell death. Competitive context is the only reason that is known so far to affect the signal sent from the receptor to the pathway. And competitive context just suggests the type of competition that takes place in the cell may affect which signaling module is signaled. In this diagram, you can see the toll-like receptors, which is just a general term for all types of toll-related receptors, sends a signal to the NF-kappa inhibitor, which in this case is IKB alpha. The inhibitor then degrades and sends a signal to the NF-kappa pathway in order to begin this process. Different signaling modules are employed in order to receive a signal from the toll-related receptor. And as I said before, it's unknown why the signal is sent from a receptor and what criteria is needed in order to describe why the receptor sends that signal to a certain pathway. But it is suspected that genetic identity and physical structure influence the pathway that is activated because this would affect the receptor and the pathway's function. In the past, it has already been found that Benzitov et al. In 1999, the toll-related receptors in signaling pathways control and induce the expression of immune response genes in order to protect the immune system and begin the cell competition process. In 1997, Janeway et al. Found that binding to the ligand spaxel affects the dorsal protein and therefore affects the cell competition process. The toll protein activates this function and the cell competition process. In 2012, Song et al. Found that physical aspects of pathways may affect the selection of pathways as it may affect the function of the type of pathway. In 2014, Johnston et al. Found that genetic identity may affect which NF-kappa factor is activated as it is dependent on function, and that signals from the toll-related receptors activate the NF-kappa factors in order to eliminate the harmful cells. So as I said before, it's unknown why the receptors send a certain signal to the NF-kappa factor, just that a receptor does send a signal to the factor. So my hypothesis will focus on the activity that it is dependent on the genetic identity and physical structures of the receptors and the pathways. In order to study this, I will first induce activity of the AMP gene, which allows for visibility of activity between receptors and pathways. To do this, I will orally infect larvae of Drosophila, which is a type of blowfly, with the Kerativora 15, which is a type of bacteria. Then, after 24 hours, the larval tissues will be examined for gene expression of the AMP gene through quantitative RT-PCR, which is a process which allows you to do this. Then binding sites between the ligands and the proteins and the receptors and pathways will then be identified. In the cells that we take from the Drosophila samples, the cells will be treated with copper sulfate and then centrifuged. And then the TUB to make a GAI4 transgene will be used to create clones of these cells. The cells that we take directly from the Drosophila sample will serve as a control group, while the clone cells will serve as my experimental group. Then activity of the clone cells will be induced through a heat shock in order to observe activity of the cells. And the process will be repeated in all types of cells. There are many types of competition, such as wild type and MYC-induced. So in this case, we'll be using wild type and minute cells, and the process will be repeated in the wild type linear cells and the minute cells. then the number of clone cells that survive the cell competition process would be compared to the amount of clone cells surviving the process in the wild-type control group. Then the cells for each type will be counted in order to identify how many survived the process and how many died in this process. The RNA will be isolated and studied in the cells from the wing discs, and then all of the RNA will be measured by quantity in order to understand the genetic identity of each cell. The growth of the cells will also be measured because the amount and rate of growth of the cells affects which pathway and which receptor is contacted. The amount of growth differs based on each pathway and cell contacted. So if we are able to identify the amount of growth and the rate of growth, you'll be able to identify which pathway is contacted by which receptor. In the past, it has already been found that NIC-induced competition and minute-induced competition use the extracellular ligand spatzel and toll-related receptors in order to induce cell competition and eliminate harmful cells by inducing pro-apoptotic genes. So we know that this is dependent on genetic identity, and it's most likely that the cells I will be studying will depend on genetic identity as well. Competitive context may also influence the NF-kappa B factor and the TAR, toll-related receptor complex, that is signaled by the factor in the complex. The specific genetic identity and physical structure of both of these components will also be affected, as well as any type of pro-apoptotic inducers used in the process, because each pro-apoptotic inducer has a different genetic identity. In this diagram, you see on the left that wild-type cells use the RPL14 pathway and contact toll-related receptors 3 and 9 by binding to the ligand spatzel and contacting inhibitors and processors in order to create cell death. While on the right, high MYC, which is a type of competition, uses wild-type cells and receptors 2, 3, 8, and 9 to bind to the spatzel ligand and use the REL factor in order to eliminate cells. In figure 2, you see that the GFP program allows you to see how many cells have died, and it shows which pathways and which inhibitors may have been used in order to do this. So you know from this information that in wild-type cells, REL, which is a type of pathway, and TOLO, which is another type of pathway, are necessary to create cell death. The graphs quantify this information. In figure C, you see that pathways are also used. Different pathways, in this case is REL68, are used to create cell death and use inhibitors and receptors as well to do this. In this case, the beta-GAI inhibitor is used in order to create cell death, and the graphs on the side quantify this information. So I believe that my research will show that competitive context does affect the cells which participate in the competition process, as well as the genetic identities of both the receptors and pathways, the physical structures of the receptors, pathways, any inducers or inhibitors used in the process, the genetic identities of the pro-apoptotic inducers, as well as any other aspects affecting the type of inhibitor and receptor used in the process. Pro-apoptotic genes are necessary for competition because it induces cell death and apoptosis in order to eliminate the harmful cells. It influences which toll-related receptor and NF-kappa B factor is chosen. Physical and genetic aspects of the NF-kappa B factors describe relationships between the pathways and toll-related receptors. Applications of my research can go towards stem cell research and cancer research, as it will allow for the understanding of why cells may grow uncontrollably and why cells may produce quickly, and understand how we can prevent harmful cells from multiplying quickly. Different signaling modules are employed in order to send a signal from the receptor to the pathway. The signal may be dependent on the genetic identity and physical structure of the cell populations that are participating in the competition process. It influences which pathway is activated by which receptor and any inhibitor or pro-apoptotic inducer used in the process. In the future, research can study if there are any variations in the cell competition process based on which NF-kappa B factor is employed, So for example, if a certain toll-related receptor contacts a certain NF-kappa B pathway, it can be studied if these two components may affect the type of competition used in cells in contrast to another type of receptor and another type of pathway. I would like to thank the family for supporting my research, my mentor, Dr. Laura Johnston, for allowing me to help her with her research this summer, and as she will be helping me with my research next year. Pauling High School for introducing the science research program, my classmates for supporting my research, and Ms. Ronaldo for supporting my research and guiding me through this process.